Hello, Biology World. The purpose of this video is to give students who couldn't get all the notes down during class, or maybe students that missed class, an opportunity to get them finished. For those of you that are doing a Google Doc and are starting the notes, you are going to need 27 to 42. You're going to need 16 rows and 2 columns. Obviously, for those of you that are writing by hand, just draw a line down the center of your page. Please remember to number them as you see them in the notes. Again, today we're going from 27 to 42. Okay, today we're talking about Mendel taking it to the next level. 11.1 and 11.2 talked about probability and Punnett squares and one-trait cross problems. Now we start getting into independent assortment and two-trait or dihybrid cross problems. And for those of you that are going to mute this video because you don't want to hear me talking, or you just want to listen to your own music, or maybe you just want silence, whatever the case may be, you will know I am getting ready to advance to the next slide when I point. Same as always. All right. So Mendel and the principle of independent assortment. Every time you see IA, that's what independent assortment is. So he wanted to find out if one pair of alleles, remember, most genes have two forms, a dominant form and then a recessive form. One pair of alleles, the segregation, remember in meiosis, during metaphase, the chromosomes get pulled apart and each gamete ends up with half of the genetic information. Whether or not the segregation of one pair of alleles would affect the segregation of another pair of alleles. And in order to do that, he formed a two-trait or two-factor dihybrid, they all mean the same thing, cross. Moving ahead. He used true breeding dominant plants. So let's go back to the one-trait cross problem. True breeding, pure breeding, same thing, okay? True breeding tall in the one-trait cross problem. True breeding short. Big T times little t, all right? Now we're using two traits at the same time. We're going to use the trait for seed shape, and really the P is the seed of the plant. Seed shape, round, dominant, wrinkled, recessive, yellow, dominant, green, recessive. So we're using seed shape and seed color at the same time. So the genotype of the double dominant, as I like to call it, is big R, big R, big R, Y, or pardon me, big R, big R, big Y, big Y. And then the true breeding recessive plant was little r, little y, little y. Now remember, in the one-trait cross problem, all of the offspring ended up with the dominant trait. And Mendel was probably scratching his head and going, I could have sworn that I had a short parent involved in this cross. So as you might expect, we're going to get a double heterozygote or a hybrid in the results. F1 offspring were dominant, round, yellow peas. They were double heterozygotes or double hybrids. All right, moving ahead. Okay, so the F1 phenotype versus the genotype. So the phenotype, they were all round, they were all yellow. They had both dominant traits. But they were double hybrids. Remember, HTZ stands for heterozygote. GT stands for genotype. PT stands for phenotype. Moving ahead. Okay, so the next steps. Just like in the one-trait cross, okay? He cross-pollinated to true breeding dominance. Those were the parents, okay? In the F1 generation, they had both, were in the one-trait cross problem. They had the dominant trait, but they were hybrids, okay? And then in the F1, he self-pollinated it. So we had the double heterozygote, as you can see here, times the double heterozygote, as you can see here. Obviously, this Punnett square is a lot bigger, All right. And that's simply because when you're talking about two traits, you have to represent all of the possible gamete combinations. Okay. So we're going to have a larger Punnett square. Okay, so the results in the F2 generation, the Punnett square predicts a 9:3 to 3:1 phenotypic ratio in the F2 generation. The 9 are going to be the round and yellow. If you count them up, you can count 9 of them. One of the 3s represents round and green, these 3. The next 3 represents wrinkled and yellow, these 3. And then the 1 represents the double recessive. Okay, out of 16 possible offspring, only 1 out of 16 is going to have the double recessive. And remember, in the one trait cross problem, when the short plant came back in the F2 generation, that led him to understand that the alleles, the two different forms of the genes, the heterozygous F1 generation got segregated or separated. And the only way for you to get, in that case, the short offspring is if a little t in the egg cell and a little t in the sperm cell, or if we're talking about plants, the pollen and then the ovule get combined. And the only way two lowercase t's can get combined is if they get separated when gametes are being formed. Same thing's going on here. All right, moving ahead. Okay, F2 conclusions, 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. All right, so the principle of independent assortment, the alleles for seed shape segregated independently of those for seed color. So when the chromosomes line up during meiosis in metaphase, they line up along the middle, the metaphase plate. The chromosomes line up randomly, okay? So that means that different gene combinations, all of the different gene combinations are possible. If the chromosomes lined up the same way every time, then we could predict fewer gene combinations. But because those chromosomes just kind of line up the way that they line up, we know that genes can be combined and end up in gametes independently of one another, which means that we have to account for all of the possible gene combinations. And let me back up just a bit. So this is a gene combination, 2 dominant alleles. This is a gene combination, a dominant and a recessive allele. This is a gene combination, a recessive and a dominant allele. And this is a gene combination, a recessive and a recessive. When I'm talking genes, we're talking about seed shape, round versus wrinkled, and seed color, green versus yellow. Okay, so because this was a heterozygote here, all right, every possible gene combination we have to account for. And that's why you have such a large Punnett square. All right, the importance. Independent assortment helps explain the many genetic variations observed in plants, animals, and organisms. If the chromosomes did not line up randomly during meiosis, during gamete formation, if there was a predetermined way that they lined up, we would not have as much variation in species as we do in plants, animals, and of course the other organisms. We're talking about fungi, we're talking about bacteria. Moving ahead. Please use this as an opportunity to practice with your notes. Mendel used a 2-trait cross to help figure out the principle of blank, blank. You might want to take a look back at number 27. You got it. Principle of independent assortment. Mendel used two traits that were dominant, round and yellow. He used two traits that were recessive, wrinkled and green. What were the results of the F1 generation? You might want to take a look back at number 28. Yeah, they were double heterozygous. They were big R, big Y, little y. They had both of the dominant traits in the phenotype, but they were double heterozygous in the genotype. When he crossed the two heterozygous, big R, big Y, little y, with one another, did the recessive traits come back? Yes or no? What were the ratios of the F2 generation? So you might want to take a look at number 31, 32. So yeah, remember, it was 1 out of 16 chance that the double homozygous recessive trait, so that would have been wrinkled and green, came back. And the ratios of the F2 generation, I know that's kind of an incomplete question. The ratios were 9 to 3 to 3 to 1, 9 having both dominant traits, 3 having a dominant trait and a recessive trait, the other 3 having the other dominant trait and the recessive trait, and the 1 having both recessive traits. Okay, do all genes show simple dominance or recessiveness? No. So you're going to learn how to do 6 types of genetics problems. Incomplete dominance basically means that the dominant trait is incomplete. It's you don't actually see the dominant or the recessive trait, or dominance or recessiveness is incomplete. This is a blend, okay? So in the case of, I think these are called 4 o'clock flowers. When you have a white flower and a red flower crossed, you get a blend. You get an intermediate, incomplete, intermediate, which means somewhere in between. All right. Incomplete dominance, heterozygote, genotype. Okay. So we get the genotype of R and W. Right? R for red, W for white. If you had complete dominance, they'd all be red. Right? But because we don't have complete dominance, the RW is pink. If you end up obviously with 2 W's, it's a white. If you end up with 2 R's, it's a red. All right. So when you get a heterozygote, it is a blend. It is an intermediate. All right. Moving ahead. All right. Codominance. All right. Co means together. All right. So co together means you will see 2 traits together. So incomplete dominance, you will see a blend. You won't see either the red or the white in the heterozygote. You will see an intermediate. Codominance, on the other hand, when you have a heterozygote, you see a speckled pattern. Example, in some chickens, both the dominant black, which is big B, feather color, and the dominant white, big W, are seen as a speckled. So obviously if you get big B, it's going to be black. If you get big W, it's going to be white. But if you get the heterozygote, you're going to see a speckled pattern, both of the dominant traits together. That's what co means. Company, bunch of people working together for a common goal or a common purpose or a common product. So we've talked about 1 trait cross problems. You have a total of 6 problems that you're going to need to learn how to do. Okay. Talked about 1 trait cross, 2 trait cross, and section 11-3, you're going to learn about 3 of the others, okay? Incomplete dominance, codominance, and multiple alleles. Moving ahead. All right, multiple alleles. When genes are controlled by more than 2 alleles. So remember, up until this point, we've been talking about dominant allele and a recessive allele. Most genes demonstrate either dominance or recessive. So 2 forms of the gene. Multiple alleles means genes that are controlled by more than 2 alleles. An example, a rabbit's coat color is determined by at least 4 different alleles. Another example, and we'll touch on this when we get to chapter 14, is human blood type. Has 3. We have the allele for A-type blood, we have the allele for B-type blood, and then we have the recessive allele for O-type blood. So 3 different alleles rather than 2 different alleles. All right, moving ahead. All right, poly is the Latin root for many. Many genes, multiple genes, okay? There are a variety of traits that are affected by more than one gene. So for example, depends on when you're watching this video, I have recorded a video of a facial characteristics lab where you have to flip a coin to determine if the dominant allele ends up in the offspring or the recessive allele ends up in the offspring. There are a number of traits in that lab that are influenced by more than one gene. Therefore, you had to flip the coin more than once. That was skin color, that was hair color, that was eye color. In humans also, how tall you are is determined by many genes. An example, skin color in humans is controlled by more than 4 different genes. So as you can tell, I mean, humans have a variety of skin tones, okay? You have, white, kind of pale, like me, and we're coming out of winter. When it gets into the summer, my skin color will change to a darker one because I have a lot of melanin. I have Greek ancestry, and Greeks live closer to the equator, and they have more melanin in their skin. Therefore, I will become darker as the sun gets higher in the sky. All right, moving ahead. Okay, what type of dominance is it when you have 2 dominant traits, red flower color, white flower color, and you can get an intermediate pink? So when you get heterozygous, it's not one dominant trait or the other, it's a blend somewhere in the middle. Is that codominance or incomplete dominance? You might want to take a look back at number 35 and 36. Yeah, that is incomplete dominance. All right, define codominance and give me an example. Take a look back at number 37 and 38. Yeah, so codominance is when you get a heterozygote, you will see both dominant traits in the offspring, kind of a speckled appearance or a spotted appearance, okay? An example of codominance is rabbit, chicken feather color. The heterozygote, you could get white and black speckled feathers. When genes are controlled by more than 2 alleles, the genes are said to be controlled by, you might want to take a look at number 39. Multiple alleles. Traits that are controlled by more than a single gene are called blank traits. Take a look at number 41. polygenic traits. Okay. In a cross involving two pea plant traits, observation of a 9:3 to 3:1 ratio in the F2 generation is evidence for two traits being inherited together, an outcome that depends on the sex of the parent, the two traits being inherited independently of one another, multiple genes being responsible for each trait. Remember, when you get all the different possible gene combinations in the gametes, that is evidence for independent assortment, or they assort, which basically means they get organized independently of one another. C. Traits controlled by two or more genes are called or more than one gene, multiple genes. Yeah, polygenic. In 4 o'clock flowers, the alleles for red flowers and white flowers show incomplete dominance. The heterozygous flowers have, so what's the phenotype of the heterozygous flowers? Remember, incomplete dominance means you will not see either dominant trait in the heterozygote. You will see a blend. And in this case, the blend will be pink. A white male horse and a tan female horse produce an offspring that has large areas of white coat and large areas of tan coat. This is an example of spotted, speckled. Actually, I think in horses it's referred to as palomino. Both dominant traits are seen. They're seen together, co-dominance. All right, ladies and gentlemen, that is it for the notes. I hope that was helpful, and we will see you in the Zoom.